today's lecture, we will discuss the next topic in molecular base of inheritance, that is 6.3. It is called RNA world. So, what is RNA world? We need to understand, right? What is RNA world and who explained this hypothesis of RNA world and who coined? Alexander Rich in 1962, right? He proposed the concept of RNA world. Alexander Rich in the year 1962 proposed the concept. He proposed the concept of RNA world. Whereas the next scientist, Walter Gilbert, in 1986, he explained the concept of RNA world. So, Walter Gilbert in 1986, he explained the concept of RNA world. Who proposed the concept of RNA world? Alexander Rich. When did he propose? 1962. Who explained the concept of RNA world? Walter Gilbert. When? In 1986. Now, what is this RNA world? So, scientists believe that when at the time of evolution in water, so what are the biomolecules which are formed? Proteins are formed and nucleic acids are formed. So, what was the first nucleic acid which has been formed in the prebiotic soup, primordial soup? So, still now scientists believe that at the time of evolution, in this prebiotic soup, this is called prebiotic soup. In the prebiotic soup, when biomolecules are made, so what are the ancient molecules which are made? So the scientists believe that in this prebiotic soup, nucleotides are first made. And at the time, the temperature was very high. The temperature was very high at the time. Since the temperature was very high, it is giving heat energy. When the heat energy is available, anabolism can happen. So, using this heat energy, the nucleotides joined themselves. The nucleotides joined themselves to make a polynucleotide, which is nothing but the RNA molecule, what we call now. Which is nothing but the RNA molecule, what we call now. So, means the scientists believe that the first genetic material which got evolved at the time of ancient evolution is RNA. So, scientists believe that. What is the first genetic material? They tell that the first genetic material is RNA only. Without any doubt, they tell the first genetic material is RNA only, and they have enough evidences also to tell that the first genetic material is RNA, because all the essential life processes has evolved around RNA, because they have evidences. They have evidences to tell that all the essential life processes what are they telling? All the essential life processes have evolved around RNA. They are telling all the important life processes have evolved around RNA. What are the important essential life processes? Like metabolism requires RNA, translation requires RNA, splicing requires RNA. So they tell that all the essential life processes has evolved around RNA. What are the processes? One, they tell metabolism. Other one, they talk about translation. And the third one, what they're telling is splicing. Now for all the three they're telling RNA is required. Now in RNA metabolism, in viruses, RNA is the genetic material. Now from RNA it will duplicate and it will make RNA, and that RNA will make proteins. So they are folded prions, they are folded particles, right? So viroids, if you see, they are RNA particles which are having infectious nature. So means metabolism means RNA metabolism. RNA metabolism, where can we find? In organisms which are having RNA as the genetic material. Not only in those organisms where RNA acts as a genetic material, even in us also, like RNA metabolism exists, and that RNA metabolism only will make the proteins. And translation, we know translation is nothing but protein synthesis. Translation is nothing but protein synthesis, and for translation to happen, we know that there is a requirement of all the three RNAs. For translation to occur, there is requirement of all the three RNAs. What did I draw? I draw ribosome. Ribosomes are made of rRNAs. So these are ribosomes. So what I am drawing inside? These are rRNAs. Now, who will copy the message? RNA will copy the message from the DNA, which comes and binds. So what is this? This is mRNA then. Then if we have to see, mRNA is required for translation, rRNA is required for translation, tRNA is also required for translation, because on the mRNA code will be there. On the mRNA code will be there. Who will read the code? tRNA will read the code. That means tRNA is also required. So this is inverted L-shaped structure of tRNA. So protein synthesis, which is called translation, requires RNA again. Metabolism requires RNA. Translation, protein synthesis requires rRNA, mRNA, tRNA, and splicing. So what is splicing? Splicing comes under post-transcriptional modifications. Splicing comes under post-transcriptional modifications. After transcription, what is transcription? Transcription means it is the process of making RNA from DNA. Transcription is a process of making RNA from DNA. This process is called transcription. Now, the RNA which is formed after transcription, is it immediately functional? No, it is not. So, it is non-functional RNA. So, that means you need to do post-transcriptional modifications and we need to make it functional. So, for post-transcriptional modifications, we will take the RNA and we will see the regions which are not required, which are non-coding, which are called as introns, and the introns should be spliced off. So the process of removal of introns and joining the exons is called as splicing. The process of removal of introns, okay, which are non-coding regions, and joining the exons is called as splicing. Splicing is mediated by a particle called spliceosome. Splicing is a process to remove introns. Splicing is mediated by spliceosome. Spliceosome is made up of snRNA with proteins. Spliceosome is a complex which is having many snRNAs combined with proteins. That means RNA is required to cut RNA. Diamond is required to cut diamond. In the same manner, RNA is required to cut the RNA. So now let's listen again. What is RNA world? Who gave the hypothesis of RNA world? So who gave the concept of RNA world? Alexander Rich in the year 1962 proposed the concept of RNA world. And who explained the concept of RNA world? So Walter Gilbert in 1986, he explained the concept of RNA world. So that means, what is RNA world? So scientists strongly believe that, means you can't have proofs because at the time of evolution, so we crossed so many billions of years and we came here. So they believe that, that's why it's a hypothesis. They believe that in the prebiotic soup, in the ancient soup, So when the evolution happened, when the biomolecules are formed, what is the first genetic material which is formed? Means in the prebiotic soup. So they tell that nucleotides are there, and at that time the temperature is very high. At that temperature, using that heat energy, the nucleotides combine with each other to make a polynucleotide, and that is the first nucleic acid, which is RNA, which is simple. Evolution also happens from simple towards complexity. RNA is simple in structure, DNA is very complex. So that's why the first genetic material which has been formed is RNA. And they tell that all the essential life processes have evolved around RNA, like metabolism requires RNA, translation or protein synthesis requires RNA, splicing also requires RNA, metabolism means RNA metabolism. RNA metabolism is very much required for organisms where RNA is genetic material. In us also, DNA makes RNA and RNA is only involved in metabolism. So RNA plays very important role in metabolism. Next one is translation or protein synthesis. So if you have to synthesize protein, it is an interaction of the three RNAs with each other. So rRNA will make the ribosomes, mRNA will copy the message, tRNA will bring the amino acids so that the amino acids are joined by a peptide bond. For that also RNA is required. Splicing, so DNA to RNA, it's called transcription. So after transcription, the RNA which is formed is not straight away functional. You need to do some modifications to make it functional. You have to identify the unwanted regions, non-coding regions, and you have to splice them off and join all the coding regions. So the non-coding region is called introns. The introns are removed and the exons are joined by a complex called spliceosome. Spliceosome is made up of snRNA combined with proteins. So RNA is cutting RNA. The mechanism is called splicing. So scientists tell that like all the essential life processes are evolved around RNA. So they tell that RNA can act as a catalyst. So they believe that RNA acts as a catalyst. There are so many biochemical reactions inside an organism which can be mediated only by RNA catalyst but not by protein catalyst. Catalyst in body is an enzyme. So enzymes, mostly we tell 99.9% enzymes, 99.9% enzymes we tell are proteinaceous. They are proteinaceous, whereas 0.1% enzymes are non-protein enzymes. They are made up of RNA. Example is ribosome. Example, one more example, RNase P. So there are some enzymes which are catalytic. There are some RNAs which are catalytic. There are some metabolic reactions, biochemical reactions where RNA only can catalyze. So we can write down that in living systems there are some biochemical reactions. In living systems, there are some biochemical reactions which are catalyzed only by RNA catalysts. Which are catalyzed only by RNA catalysts. Means only by this 0.1 fraction, not by 99 point fraction, right? So that means RNA is catalytic. When RNA is catalytic, it means it is reactive. If it is reactive, means it is unstable. Since RNA is catalytic, catalytic means RNA becomes reactive. RNA is more reactive. If RNA is more reactive, that means RNA is unstable. It is reactive means stability is not there. RNA is unstable. Now, if RNA is unstable, we told stability is an important point for genetic material. So, RNA cannot act as a genetic material for most of the organisms. So, then DNA has evolved from RNA. That means RNA has made some chemical modifications and got converted to DNA. So, we can tell, therefore, DNA has evolved from where? Has evolved from the first genetic material from RNA. That means RNA has undergone some chemical modifications and became DNA, and DNA became stable. So we can tell therefore DNA has evolved from RNA with some chemical modification. So the RNA, children, has made some chemical modifications and got converted to DNA, and then DNA became stable because some chemical changes it made. Those chemical changes have added stability to the DNA. So again, RNA is single stranded, whereas DNA is double stranded. Now this is the sequence present on the RNA. Now if any mutation happens on the RNA, like these bases got converted into AA, mutation happened. X-ray radiation, mutation happened. You'll not even come to know that mutation happened on the DNA. Whereas in RNA, since they are double stranded, now it is G here and it will be C here. Now if mutation happens here, if G becomes AA, then opposite CCs are there. On the opposite, mutation happens on one strand. So then GG is like here, it got converted to AA. Now we can know that there is a mutation here. How do you know that there is a mutation here? Here we don't know that mutation happened because of its single stranded nature. Now because of its base complementarity, we are able to know that there is a mutation. When there is a mutation, then DNA has evolved a repair mechanism also. DNA can do repair mechanism also. It can do repair mechanism also. So it knows that my original bases are CC, because of mutations AA came. So now repair enzymes will come and they will add GG. Or else these two bases will resist to mutation, will resist to mutagen, and they will not allow them to change. So we can tell that DNA resists mutations, where RNA cannot resist mutations, and RNA can easily undergo mutations. So here also we can tell that why RNA got converted to DNA. Since it should be the genetic material, for genetic material stability is required. RNA cannot be stable. Why? Because it is reactive, it cannot be stable. Because it is catalytic, it is reactive, it cannot be stable. That is why RNA with some chemical modifications became DNA. That too, single stranded RNA became double stranded DNA. Since it is double stranded, now at one point if mutation happens, the other bases can help in repair and bring back the original bases. Okay, DNA has evolved repair mechanism. And to again support RNA world, we can take the concept of Miller's experiment. Test tube experiment we will tell. Miller's experiment means, so scientists believe that the primordial soup, these components are there, it's a very high temperature. At that high temperature, nucleotides are joined. So Miller conducted experiments in his lab. Miller conducted experiments and he proved the theory of chemical evolution. Now, the Miller's experiment also showed that when he has taken the nitrogen, carbon, hydrogen, oxygen, and when he maintained 800 degrees centigrade and anaerobic conditions, he also saw that proteins, amino acids and nucleotides in RNA has been formed first. So, this Miller's experiment also supports RNA world. It also supports RNA world. Viroid concept also. Now, viroids are discovered by T.O. Diner. Viroids are discovered by T.O. Diner. Now, what are viroids? Viroids are small, low molecular weight RNA molecules without protein capsid, right? They are naked. They don't have a protein capsid. They are just low molecular weight RNA molecules, and we tell viruses are primitive, ancient than bacteria and all. So viroids, which are having only low molecular weight RNA molecules, so they are capable of causing diseases. They are capable of causing diseases. Means this viroid, when it takes a host, when it enters into a host, so it can use the host machinery and it can cause disease to the host. So children, viroid concept is also supporting RNA world. Miller's experiment is also supporting RNA world, and viruses actually, so some scientists believe that viruses are the connecting links. Viruses are the connecting links between non-living things to living things. And majority of the viruses will have RNA as the genetic material. And then like origin of sexual reproduction also has evolved around RNA. So when we are telling that in the prebiotic soup, when RNA is made and RNA is covered by a protein coat, now it behaved like a cell, it behaved like a protobiont. It behaved like a protobiont. This protobiont has generated the replicating capacity. It has generated the replicating capacity. It has generated the reproductive capacity. And it started doing sexual reproduction to increase the number of cells. So, origin of sexual reproduction also happened with the RNA molecules only. And one more concept is there. Biocoid is a type of RNA which they have recently discovered in the Drosophila egg. Which they have recently discovered in the Drosophila egg. Biocoid is nothing but the RNA which is there in the Drosophila egg. So this is a RNA which is present in the Drosophila egg is called as biocoid. Now when this egg fuses with the sperm, when this egg fuses with the sperm, now then this biocoid RNA will make the genes which are present here active. The biocoid will make the genes which are present here more active, and then it makes the embryo, then this will make the head of the Drosophila, and the genes which are present on the opposite, the biocoid RNA, it will make the genes which are present at the tail less active. Because they are less active, they are shrinked and they'll make the tail region. And because the biocoid RNA which is present towards this side, it is more active, it will make the head and it will make all the sensory structures. It will make all the sensory structures. Now also we can see that RNA is having so many actions, and RNA is an advanced particle than DNA. So that's why we tell RNA is dynamic. So with all these examples, we can tell that RNA is the genetic material and RNA has many roles. RNA is dynamic, and this is a concept of RNA world. These are the explanations what Walter Gilbert in the year 1986 he explained about RNA world. So children, we have studied about DNA, we have studied about RNA. Now when we come to the main process of molecular biology, it comes with replication. So in the next class only, not in this session, yes, not now. In the next session, we will discuss about the central dogma of molecular biology proposed by Crick, and we'll take up the first part of molecular biology, replication, and we'll start. So hope you're understanding the lectures. If you have any queries, you can post them in the comment box so that it will help me to understand and then work out in a better manner. If you like the information, like, share and subscribe to my channel. Thank you.